Thank you. Good. I was really optimistic over the time, so I prepared more slides, but then I'll cut it down. EMPE Diagnostics is a R&D based SME that focuses on developing point-of-care diagnostics. And our aim is to give the appropriate antibiotic treatment already in the first clinical contact. I am the co-founder and CEO of EMPE Diagnostics. So I'll skip all these slides because of time. Tuberculosis is a major problem, as everybody knows. And 2.5 billion people are infected with tuberculosis. And at every 20 seconds, one person is being died. The treatment is so long with multiple antibiotics. It takes up to 36 months for treating one multi-drug resistant patient. The same patient could look like this after the severe infection. But if we can have a potent as well as a proficient diagnostic test that can give the confirmatory answers already in the first clinical meeting, then basically we can push down the diagnosis time as well as bring down the cost. Most of the TB cases are treatable with the currently available antibiotics, but if we cannot identify or treat it properly, then the multi-drug resistance will shoot up and we will never, or most probably we will never treat it in the future. So that's why WHO says that we have to develop a process where we can diagnose the tuberculosis at the very early stages. There are so many, or not so many, there are a few diagnostic companies are available today, but providing services, but all of them requires high infrastructures as well as labor to use. But we want to position ourselves in the primary healthcare centers. Our solution is we combined a molecular test together with a lateral flow strip, and we made it as a very simple, like a pregnancy test. And so I have the prototypes for the cassettes if you want to see. And we wanted to bring down the diagnosis time as well as the treatment cost to $500 with diagnosis time with 90 minutes. We have established a clinical network with 16 countries, including the 7 WHO national reference laboratories across Europe and South America, where we are actively conducting our clinical validations. So we recently got our first clinical validation report. Okay, there is something missing. I also put the sensitivity and specificity issues here. But then, as you can see here, there is no necessity for external report for the doctor. So as you can see here, NH plus says that it is like isoniazid positive. This patient can be treated with isoniazid. If you see the signal here, then we can say that this patient cannot be treated with isoniazid. But if you see the signal here, this patient is responsible, can be treated with rifampicin, and if you see the signal here, then the patient will not respond to rifampicin. So it has the 100% specificity and 100% sensitivity. So we have the next product which can identify antibiotic resistance or genotype all the antibiotics available today. And we are also working on developing a completely automized instrument for the identification of tuberculosis. We have been glad to say that we have been identified as one of the startup companies in the Swedish landscape of precision medicine. And we have recently got a couple of projects, including a European Union project. And we have been identified as one of the top 20 medtech solution providers in Europe. Last year I was here, I was meeting all the people, and we are closely discussing with the government in order to bring our test kits to India. And now we have established a clinical consortium in India with 15 clinical partners. And we wanted to develop one test kit that can address all the TB patients, irrespective of the geographical variations in 2019. I have been invited to present the concept in a WHO conference in next week. Thank you. I'm in time.
You mentioned that one of the products has got clinical validation.
Yes.
Have you started commercialization at any market yet?
No. So the first phase of the clinical validation is over. That means in Spain. We wanted to really do a mass clinical validation before we can get the WHO approval.
What is the expected price?
Yeah. So yeah. So now we have forecasted the price according to the Swedish manufacturing conditions, but we are establishing a production facility in India so that we wanted to match the Indian government needs. So we will be in that pocket. Yes, very affordable. Okay, so we have the two tests. One is the MDR and the other one is the XDR. So this one we suggest it to perform on sputum positive microscopy samples, that is 3+ microscopy positive samples. And we are working on this one. This can be applied on the sputum samples itself. Basically, we wanted to develop a system that can replace microscopy, which has the 50% false positives.
So you mentioned that you are in phase 1 right now for clinical evaluation. Yeah. So you'll and you have 15 sites currently going on only in India. So is there a plan that is drafted for phase 2 and phase 3 and also phase 4 of clinical trials?
Yeah, so this is little bit different. I mean, I think we are talking about the pharmaceutical clinical trials, and this is little bit different when it comes to the diagnostic clinical validations. Right. So we don't need a clinical trial here, but we need a clinical validation. So that means that it is one step. So right. we when the diagnostic, yeah.
Right. Okay. So yeah, makes sense. So, but still, this will be probably a class 3 product. Since it belongs to tuberculosis testing?
You're absolutely right. So that's why these two diagnostic tests, like MDR-TB and XDR-TB, these cassette, that requires a small amplification of 40 minutes and also a sample preparation. So we suggest them to be carried out in a laboratory where they have the facilities, in order to satisfy the biosafety measures. But the automated machine, which I was showing here, so. Okay, anyway, so the automated machine, which is a black box, it has all the components of sample preparation as well as the amplification and also the readout. So that can be placed in the primary healthcare centers.
Okay, yeah, sounds good, thanks.
So you mentioned 100% sensitivity and 100% specificity.
Yeah.
What is that against?
So the technology has a single molecule precision, basically. Theoretically, we can identify even a single molecule in a complex system. So when we have validated this one in the lab as well as in our, with the clinical samples, we could see 100% specificity. And there was a table where I have compared our concordance of this method with the pyrosequencing as well as other molecular technique. So unfortunately, I could not show this. It is, somehow happened.
No, but so you're saying that so those techniques are gold standard. They don't have any false positives, false negatives whatsoever. Yeah. The sequencing
sequencing. So because I'm telling that we have verified the samples across 2 different sequencing sets, and then we used it.
And what was the sample size?
We have done the initial validation on 25 samples. Okay. 25 samples.
And how many positives and how many negatives?
So in this one, 9. So okay. So among these 21 samples that have been sequenced, which we obtained from the Swedish WHO National Reference Laboratory, and all those samples were verified by them, and we have been validating in our lab. So one of the samples, so in this case we have like 4 genes, 4 codons, and among those 4 codons, we could not identify only one codon of rifampicin positive. That led us a specificity value of 87.5% for the rifampicin rifampicin mutant. This is because that codon was overlapping with other gene, other codon because of the SNPs. We are covering that mutation in our next in our XTR TB product.
Okay.
Yeah. So, sorry.
What's the underlying technology here?
So it's called like padlock probe and rolling circle amplification technology, which is an isothermal amplification method, similar to. So we use. So we represented the logo as here. So it represents the padlock probe, attaches to the target, and then after that it forms
Sure.
a circle. And then
Sure.
rolls.
So, but all this is thermostable, right? Because Indian
Yes.
environment is very, very different from
Yes.
what you see in
Yes.
other parts
Yes.
of the world, right?
Yes, we do the
So,
amplification
yeah.
at 37
Because
degrees.
there are a couple of companies which have come into this, and your own slide indicated about MolBio, but it hasn't really created that impact.
Yeah.
The impact that one can possibly create out of this is huge.
Yeah.
But I really fail to understand why things haven't worked out the way
Yeah.
everybody expected. So that's something that you need to look into.
Yeah, yeah. Because like, so I don't want to comment about
So I
those.
think we can
but
connect offline in interest of time, so we can connect
yeah,
offline.
yeah. So we strictly follow the WHO guidelines, and I think we can achieve that.
Yeah, just a last comment more than a question. So the Global Fund for HIV, TB and Malaria has launched the India Health Fund last year. And now they are looking at TB diagnostics space, and they are looking to sort of fund some innovative ideas and bring them to India. So I think you should definitely look at that.
That
And
would be-
please connect with me to understand how we can help you bring your products to India.
That would be wonderful.
Yeah.
Thank you so
Thank
much.
you.
Thank you. I appreciate it.
All the best.
Thank you.
Thank you, Mr. Pavan. So may I now request for the third and the last Health Cubed India Private Limited to please present their presentation over here. Thank you.